क्या करें जब उसका ट्रैवल मॉड्यूल समझ नहीं सीए के लिए अप्लाई कर रहे हैं तो लिखा कि आर यू ट्रैवलिंग ओवर चेक करी वही कल ये लोग कुछ भी पूछेंगे भाई आपको
और क्या पूछेंगे यार बात
मैंने इनको पूछा मैं आप बाकी क्या बाकी कुछ और पढ़
So welcome. ये ऑडियो किसका आ रहा है? Whose audio is coming? Whose audio is coming? Okay, so we have talked about restriction finder. Anything that you would like to ask about the finding restriction sites? Uh, so it means that you can easily find restriction site yourself. So if any query came that you can, the main thing that you need site finder and choosing your enzyme of choice. Okay, so now we can move to the our next part, that is performing our experiment. So how to perform it? So for the performing experiment, as I mentioned that uh, depending on the requirement, you have to choose your enzymes, what enzyme you need. So then you have to mix your ingredient. So then what different ingredients are there? What you need? You need DNA, and DNA is a solution form. So you need liquid DNA liquors. You must know the amount. So if you want to, generally in case of our experiment, we use to digest our plasmid. You need a plasmid DNA, or if it is a genomic DNA, then you need genomic DNA. You must be aware about how much amount of DNA you are having, because amount of DNA is going to tell you how much enzyme you have to use. Then you need a restriction enzyme that should be appropriate, means what restriction enzyme you want to use, where you want to digest, what type of fragment you need, and with the restriction enzyme you need appropriate buffer, because if you are going to change the buffer, that will lead to uh, star activity things, means activity of your enzyme may change. So your buffer must be appropriate, means generally when you buy restriction enzymes, company provides you with buffer also with the restriction enzymes, and these buffers generally came in the form of 10x way. So whatever volume you want to use, so accordingly you can add, like your 10 microliter reaction is there, then you need to add 1 microliter of your buffer, so that final concentration is going to be 1x. Then your loading die. So loading die you need for electrophoresis to check whether the digestion is complete or not. Then you need to for the electrophoresis buffer. So buffer is also the job of the electrophoresis pipettes and tips that you all need. Equipment wise, what you need? So equipment wise, just for the sake of pasteurization, you only need your pipette man and place for incubation, or your incubator, or simply you can say water bath, where you can place your enzyme within your time, or if you want to, if you incubator you can say. So you need incubator to incubate your mixture. So how to proceed? So select your resting enzyme that want to digest the plasmid. So you need to make a reaction mixture. So reaction mixture you can make in a form of a tabulated form. So these are the things that you need to add. So as I mentioned that select your restrict enzyme, determine appropriate reaction buffer by reading the instruction for your enzyme. In a 1 to 0.5 ml tubes, combine the following. So just 1.5 ml tube you need to take, to which is called the microcentrifuge tube also. Just make a table, and this is to check how much you have to add and how much volume you need to go for. So DNA we need to go for 0.5 to 2 microgram. DNA you need. Restriction enzyme you need to in the international unit form. So as mentioned, 1 microgram you need for 1 unit. So for above setup you need 0.5 to 2 unit. Then it is going to be IU. It is IU international unit. Then come to the buffer. So buffer, that is going to depend upon what final volume is going to be. So if your final volume is going to be, say, 50 microliter, then buffer is you need to go be 5 microliter. Then comes with the water you need to add. So water you need to go for a total volume. So total volume is 50 microliter. So like if you say the 2 microgram your DNA you are adding, so 2 microgram DNA, what volume it is going to be there? So for say this DNA is going to be present in 20 microliter of your volume. So then 20 microliter is this one. This unit is going to present in 1 microliter. Okay. So this is also going to be the volume that you are going to add. This is 1 microliter. Then this is the 5 microliter buffer that you have added. So the total volume 20, 21, and 5, 26. So 50 volume is the final. So you need to add 24 UL of your water. So in this way you have to mix it. This is called the restriction digestion mix or the reaction mix. Mix gently by pipetting. So why we are saying gently? Because if you are mixing them rigorously, then shearing may also occur. Shearing means that is not the action of your restriction enzyme, but your mechanical action. Your DNA is going to be broken down. So you need to mix gently. Then incubate at appropriate temperature, that is 37 degrees Celsius. Usually because most of enzymes have a temperature optima of 37 degrees Celsius. Incubation time is going to be 1 hour, as we want complete digestion, so that's what we need 1 hour. And if you want to go for a partial digestion, in that case we can standardize your time, how much partial digestion you need, like 5 minutes, 10 minutes, 20 minutes, 30 minutes. So different time you can standardize, and that is going to be done. Then come to some precaution that you need to follow while you are performing this experiment. All steps should be carried out at ice, except the final incubation. Why we are saying you should do it ice? Because we are doing with working with the enzyme. One thing, enzymes are going to be sensitive to the temperature. And second thing, as you bring down to the room temperature 37 degrees Celsius, enzymes start functioning. Store the digested sample in freeze condition till you are using further, because you are digested one and you are going to have nucleus. So any change in thing may lead to your over digestion also. Then you should be free from contamination. This is a universal protocol when you are working with DNA. You can't bear the contamination because they may digest, may lead to no specific factors in your DNA. And next thing is what type of digestion you are performing. Like single digest, you are digesting your DNA with one enzyme, double digest, I think, 2 enzymes, triple digestion, or so on. With how many enzymes at a time you are digesting. Generally, when we are performing our experiment, we go for the single digestion. At a time, we are performing one restriction digestion. But in many cases, we need to go for the double digest also. Double digest means 2 restriction enzymes you are using for that digestion. In that case, as I mentioned, that they are very sensitive to your buffer. So only those 2 enzymes you can use at the same time, which are compatible. Their buffer are compatible. Otherwise, you can't use. Okay, so you need to check what are the buffer conditions at which both of the enzymes are working. Okay, so in that way, we are not working so much on the restriction enzyme in a research type of way. We are not standardizing the buffers. So for that, the companies that are supplying to the enzyme, they also provide you a compatibility chart. Compatibility chart means the buffer that you are having at this condition of the enzyme that you are having, which another enzyme you can use for the double digestion. Then we come to the triple one. So triple one is going to become much more complicated. So we rarely do a triple digestion at the same time. But we want to digest this with multiple enzymes. Generally, what we do first we digest with one enzyme, then we extract our DNA, then we digest with the second enzyme, followed by the third enzyme. This is the thing that we are doing in restriction digestion. So once you are mixing your all the ingredients, so your reaction fluids, your reaction mixture will pollen, and your reaction digestion start immediately. Okay, so optimal temperature for 1 hour or longer time that you need to standardize. Uh, all that everything is dependent upon what your enzyme is there, what your conditions are going to be there. Once you standardize your time, then that leads to that. Otherwise, if you leave over that time, then may lead to a non-specific degradation of your DNA. So that's why it's must when your incubation time is over, immediately transfer your sample to the ice. Or once your restoration is completed, then directly go for the agrogel electrophoresis. As you perform the electrogel process, all the fractions are going to be separated. So we need not to go for a non-specific digestion. Okay. So this is a thing, and here you can see the example how what things are going to be there. Here you can say that this is a plasmid backbone of 2870 base pair. So here you are seeing gene of interest is there, that is of 200 base pair, and it is having 2 eco R1 restriction site. So now if you treat this plasmid with restriction enzyme, so what type of fragment you are going to get? What are the probabilities of fraction? How you are going to analyze when you run your DNA on your gel? So, like this is a fragment which is 2870. So when you run this 2870, so you can say that in this case, when it is uncut means no restriction fragmentation. So when this uncut form, then at what position it should run? When it is uncut, this is your DNA, plasmid DNA. Its size is 2870. When it's uncut, where it should run? So this size is a later. You can see this is a later one. So it is having these are the sizes. Approximation 200, 300, 500 is going to be here. 700, 1000, 2000, 3000, 4, 6, 8, and 10. This is the one. So if there is no restriction digestion in your DNA, where it should run on your gel? At what size it should run? Size of your plasmid is 2870. Devanshi? Devanshi? Maniswini?
Yes, sir.
Yes, Manashwini, I'm asking a simple question that if we are performing a restriction digestion experiment, and in the restriction digestion we mentioned that where it is going to produce a cut, it will produce a fragment of that size. We started with the plasmid, and plasmid is of size 2870 base pair. When there is no cut is there in the plasmid, then where should it run?
Sir, won't it will move maybe with a low mobility and less speed, but won't it still go towards the positive terminal?
No, I'm asking this is a result. Okay,
So
this is
then,
gel process you
shouldn't
have. So there is fragment location that are there in it. So your plasmid is uncut, no cut is there in that one, and plasmid size is 2870. Then where its location is going to be? Okay, so simply just I am again moving to your whiteboard, so there I think you will get understand better. So this is your gel, we can say. Here is a negative one, and here is a positive one, and these are going to be your well from where we are going to start. Okay, and what I'm saying, we are having a plasmid. The size of plasmid is 2780. Okay, this is the size of it. This is a plasmid, and it has two recognition sequence for the carbon. This is this one, and here also. And this segment between these two sides is going to be 1200. Okay, so this. So total size is going to be 27. Okay, so simple calculation. Just simply you can get, it's not just, it is just 2700. Okay, so in the first we run our ladder. So now I'm making what are the fragment size of the ladder are there. This is 6000 or 8000, 8K, then 6K, and 4K, then 2, then 1, and then this is a 0.5. Okay, so these are the different fragment lengths. These are a ladder strand that we are saying if you run any DNA that are going to be there, that are going to move from negative to positive. So if I'm just running this plasmid, which is a 2700, just this plasmid is there. I just load this plasmid. I've not treated with this restriction engine. So where the band of this plasmid should be, where this plasmid should run, and what type of pattern this plasmid should give. It is 2780, so it might run, I think, here. It should run here. This is this is 1, 2. Yeah. This is sorry. This is 1. This is 0.5. 1, 2, 3, 4. So it should run here in between. I'm right. I'm right. Am I right?
Yes.
कोई जग रहा है? सभी सो गए? Everyone slept or anyone is waken also? All are sleeping?
No, sir.
No, sir.
No,
Okay, तो कौन कौन जग रहा है? Please raise your hand who are alive. Yes, Yukta is there. So Yukta, are you able to understand what I'm saying and what I'm asking?
Yes, sir.
Okay, so where this plasmid, where should the location of this plasmid be? So this is the location I mentioned that when you're running your gel, yes, Yuvraj, you want to answer? Yuvraj? Yuvraj?
Sir, shouldn't it be a bit more towards the positive?
Means it should be here.
No, sir, it's a little bit up.
Here, positive, this is negative.
Yes, sir, around that.
This location is below 2 or near 2. So what? So should I make it here? Yukta?
No, sir, you erased it. How should I say
Okay,
this?
just tell me at which point I should make it. Just a clear one so you can better way. Okay, so this is going to be a 0.5. This is going to be 1. This is going to be 2. This is going to be 3. This is going to be simpler one in my writing. 4. This is going to be 5. This is going to be 6. A simpler one. Okay, so your size is 2.7. So this is 2.7. If I say this is a 2, this is 3. So 2.7 is going to be a little bit below 3. It is a 2.7. Okay, so am I right in this case? So when you're
Yes,
running
sir.
this plasmid on the electrophores, it should run at this location.
Yes, sir.
Okay, so all are agree with it? What Yukta is saying?
Yes, sir.
Hmm?
Yes, sir.
And if I say no, if I say no, you all are wrong. कोई और विरोध करना चाहेगा कि मैं कह रहा हूँ आप गलत कह रहे हो? So anyone would like to object on me? No sir, you all are right. कोई बताएगा? या तो आप लोग ध्यान नहीं देते हैं जब क्लास चल रही होती है. पिछली क्लास में इतना जोर दे के मैंने कुछ चीज बताई थी, पूरी इमेज भी दिखाई थी, सारा चीज भी बताई थी, पूरा ड्रॉ करके भी दिखाया था. व्हाई इट इज हैपनिंग? प्लाज्मिड इज सर्कुलर and circular one may be supercoiled, open circular, and neck one. So different types of things are going to be there, isn't it? Is it or not?
Sir, so you are saying we are right or wrong? I still don't get it.
You all are wrong. Circular
So,
DNA
sir, where
runs
it should be?
slower than your linear DNA. So it should be at this location at a higher size location. If it is uncut, however, actual size is 2.2700 base pairs, but it is going to run at a lower speed, at a higher position. Okay, so this is because supercoiling is there. The circular one form is there. Some resistance is there in their migration. You can't change its shape, so that's why it's running at a higher speed. Sorry, it is running at a slower speed or higher molecular weight. Then come to your restriction experiment. So two possibilities are there. One possibility, as I mentioning, that a partial cut. Partial cut means only one side is cut down. So if it is cutting at only one location, then what is going to happen? Like either E1 or E2. One of these two position it is cutting, like eco R1 is there, any one of these it is cutting. Then what will happen?
So then won't it uncoil and then it will move lower?
Then it becomes linear.
Yes, sir.
It's become linear, and the size is still there 2700. So that moves at its actual speed at per second 2.7. So in this case, when it is a single location is going to digest and at one location it will digest, then it will produce a band which is going to come here below 3. So this is going to be 2.7. Now another probability is there. So this is one cut at one location. So this is going to become linear. So now another thing is there. Now possibility is there. It produce cut at both the location, both the sides. Then what is going to happen? If it is cutting at both the side at this location also and at this location also, then
So then won't we get two fragments?
yes, then we are going to get two fragments. One fragment is going to be of what length? What are going to be the length of these two fragments? What are the length going to be these fragments? साधारण गणित का सवाल है तो सिंपल मैथमेटिक्स जो कि
1250.
yes, one is going to be 1200. This fragment is going to be of 1200, and this fragment.
But
is
sir, I had a doubt. Didn't we learn in the last class that the circular will move faster than the linear one? You just mentioned the opposite, so I'm confused. That the linear one will move faster. You just said.
I'm saying in the previous slide, if you just I will show you. I will show you those things that you are asking. Just okay. Okay, so two fragments are going to be there in this case. One is going to be 1200. So this is one. So just above this location. Another is of 1500. That also going to be down here. Now another possibility is there. Some DNA where it is cutting at the both the location, and somewhere it may cut at the one location. Then what is going to happen? If some plasmids are being cut it down at the both the restriction sites and some at only one site, then what will happen? Yes. If it is cutting at some of the plasmids are cut it down at the both the location, some cut at the one location. So in that case, we are going to have three bands. In the plasmid, in which one side is being cutted, then it becomes linear. Then the size of 2.7, that is this one. And in those it is cutted at both the locations, that is going to show this two. Okay, so this is a fragment analysis you can do. By this way you can check and you can tell that how many restriction sequences are there and what are the different locations and what are the probability of this is going to be when it is going to be uncut, and this type of band is going to be there when it's linearized, then this size, and by the size of linear one you can tell this is the size of your plasmid. By the fragment you can tell them their location. Okay, and now to the answer of Yukta. She asked when I were talking about your plasmids. I mentioned that different types of speeds are going to be there. For that, you should. That's why I asked you the location where it is going to be. So this is the one. So if it is
Yes,
nicked,
sir.
if it is nicked, then it is going to be at a higher side position, and it is going to be supercoiled, then it is going to be at a lower position. So both the type of the DNA are going to be there in your sample, nicked one and the supercoiled one. So in that case, you are not going to have a single band. That's why I didn't make two bands there. So in that case, what is going to happen? We are going to see yes. So in that case, you are going to see two bands. One is going to be at higher location, other should be at the lower location. Okay, so this will tell you that your plasmid is uncut, single band. It means at a cut at a single location, and two smaller sizes band. It means that it is completely digest. Three band. It means it is partially digested. And so the bands are there in which it's cut at single location and the both the locations. Okay, so Yukta, that is the reason I made only one band to check whether you are a bit aware about the second band or not. Okay, you are right that
Okay, sir.
circular DNA, when it is supercoiled, it is going to run a bit slower, but when it is open circular, then it is going to run slower. This slower
Okay.
that I made, that is of open circular.
Hmm.
Okay, means uh, your supercoil is being removed. And when we treat your DNA with ethylene bromide, here I am telling you why I made this one. So when we treat this with the ethylene bromide can remove the supercoiling. So it makes from negative to the positive supercoil. So in that case, if it become open circular position, then you may get one band also, and that one band is going to be at a higher location. Okay.
Yes, sir. Yes.
Okay, so that is the thing, and with that, this is the way how you are going to perform your restriction digestion experiments. I will show you some video to have an idea so how really you are doing and what type of things that you will get. Just wait for a moment. I'm sharing you some videos. Okay, hope video is available to be visible to you.
Yes, sir.
Okay, so this is how to perform the restriction digestion.
Restriction digest and flag the DNA. In this activity, DNA from bacteriophage lambda will be digested with PST1, eco 4 color-coded microcentrifuge tubes with L for lambda DNA, E for EcoR1, P for PST1, and H for Hindi3. Label all of the tubes with their initials and date and place them in a microcentrifuge tube rack. Start by loading each of the colored tubes with lambda DNA. Using a fresh tip, transfer 4 microliters of lambda DNA from your stock tube to the color-coded L tube. When obtaining your sample, work at eye level so you can be certain your pipette tip is in the sample. Closely at the tip to make certain you have obtained the correct amount of sample. Then transfer the sample to the matching color-coded tube. Place the tip near the bottom of the tube at eye level to make certain all of the sample has been transferred. Continue this procedure for transferring lambda DNA to the other color-coded sample tubes. Once you have finished transferring 4 microliters of lambda DNA to each of the color-coded tubes, reset your micropipette and use a fresh tip to transfer 6 microliters of 2x restriction buffer to the color-coded tube labeled L. Again, using a fresh tip, reset your micropipette to transfer 5 microliters of 2x restriction buffer to the color-coded tube labeled E. Continue this procedure using the same volume of 5 microliters to transfer restriction buffer to the tubes labeled P and H. Once you have finished transferring 2x restriction buffer to each tube, reset your micropipette and use a fresh tip to transfer 1 microliter of restriction enzyme to the corresponding tube. When you have finished placing restriction enzyme into each tube, mix the components by gently flicking the tubes. Then place the samples in a microcentrifuge, making certain the load is properly balanced. Spin the samples for a few seconds to collect the samples at the bottom of each tube. After spinning the tubes, place the tubes in a rack and incubate for 30 minutes in an incubator or water bath at 37 degrees Celsius or overnight at room temperature. After the samples have completed their digest reactions, they are ready to be loaded and run for analysis. Please see our video for loading and running an agarose gel for help with this procedure.
Okay, so this is the way how you perform the restriction digestion, and after completing the restriction digestion, how to analyze your gel, and this is a simple thing. And if you are going to be in the lab, then we can perform this experiment here, but being online, this is the way we can do. Okay, so if you have any query regarding this experiment, we will discuss in our next class. Now we have to end our session, so please tell me, Vivek, and was the next was absent. Vivek and Tridev. 3 students are absent now. So Vivek, Tridev, these are 2. And who is the third one? Who is the third one absent? Kshitij is coming on and off. So Demetrius.
Present, sir.
Manaswini,
Present, sir.
Adish,
Present, sir.
Yuvraj,
Present, sir.
Sanjana,
Present, sir.
Nehal,
Present, sir.
Atoiba,
Present, sir.
Devanshi,
Present, sir.
Nandini,
Present, sir.
Kshitij,
Present, sir.
Janvi, Janvi is absent. Okay, Janvi was absent in previous class also. Okay, so thank you all. Stay safe, stay happy. Okay, so Janvi, 3 students are absent. Janvi, Tridev, and Vivek. Okay, so thank you, students. Stay safe, stay happy. See you again on Friday with our next session, in which we will continue with our theory. Okay, so stay safe, stay happy. Good day.
Thank you, sir. Thank you, sir.
Thank you, sir.